My name is Karen Binder, and I'm the Assistant Director of the Investigative Genetic Genealogy Center at Ramapo College. Today I'll be presenting what is investigative genetic genealogy. This video will provide a brief overview of what investigative genetic genealogy is and how it can be applied to cases of unidentified human remains and violent crimes. You may have heard of investigative genetic genealogy as it is a groundbreaking and newsmaking tool for resolving unidentified remains in violent crime cases. But how does it work? First, investigative genetic genealogy, or IGG as I'll call it in this presentation, goes by many names. You may have also heard this field called forensic genetic genealogy or just forensic genealogy. IGG utilizes advanced DNA testing techniques along with traditional genealogy and genetic genealogy research to develop investigative leads in violent crime cases and unidentified human remains cases. IGG entered the public's perception in 2018 when a number of long cold cases were solved with this new technique. IGG research began in 2017 and was performed by several pioneering practitioners, each of whom are leaders in the field today. In April of 2018, Marcia King, formerly known as Buckskin Girl, was identified by the DNA Doe Project, led by Colleen Fitzpatrick and Margaret Press. This proved to the public and to law enforcement agencies that long unresolved cases of unidentified human remains could be solved with IGG. Just 2 weeks later, Joseph DeAngelo, known as the Golden State Killer, was arrested after Dr. Barbara Ray Venter led an investigative genetic genealogy team to identify him. This catapulted IGG into the public consciousness. In July of 2018, John D. Miller was arrested for the abduction, rape, and murder of 8-year-old April Tinsley in Indiana. C.C. Moore and Parabon Nanolabs had conducted the IGG work in this case, and the 59-year-old was sentenced to 80 years in prison. As a result, he will never harm another child. C.C. Moore also led the IGG work, which contributed to the exoneration of Christopher Tapp in 2019. This was the first time that IGG had been utilized to identify the true perpetrator in a case of wrongful conviction. These cases paved the way, but since they were solved, more than 1,300 cases have been resolved using IGG across the world. This tool is the most significant and powerful development in the resolution of violent crime since the discovery of DNA itself. While the use of DNA to identify perpetrators of violent crime and identify human remains is not new, IGG utilizes a more advanced type of DNA testing which has not traditionally been used for investigative purposes. Traditional DNA testing involves analysis of STRs or short tandem repeats. You can think of this type of testing like a grainy photo on your 2006 flip phone. It's useful for capturing a picture, but not picking up the finer details. STR testing uses tens or dozens of markers along the genome and can be used to compare a subject to their own sample or to a sample from their direct relative, such as their parent or child. SNP testing, or single nucleotide polymorphism testing, is a type of testing used for the development of profile suitable for IgG. This type of DNA testing examines hundreds of thousands or millions of markers along the genome. Think of this like a high-definition photo on your smartphone in today's world. Great for zooming in on the finer details. SNP testing can be used to identify not only the DNA contributor and their direct relatives, but also their third, fourth, fifth, and more distant cousins. Development of a SNP profile depends on advanced DNA testing from a qualified laboratory. Next, I'll provide an overview of the lab process that precedes IGG. IGG practitioners are typically not experts in forensic DNA, although there are exceptions, and thus they rely on relationships with forensic DNA laboratories to guide the laboratory process for the successful development of a SNP profile. The process begins with specimen collection, wherein the investigating agency and laboratory select the appropriate specimen for testing. Commonly tested specimens include existing DNA extract, bone, teeth, hair, including rootless hair, blood, semen, and more. Next, extraction is performed to isolate the DNA of the individual the agency wishes to identify. If you've ever seen a child complete a science project where they extracted DNA from a strawberry, you'll have seen what DNA extract looks like. After that, genotyping is the next step. This is where the sequence of the individual's DNA is analyzed to develop the genetic profile for the subject. This can be done through various methods, including whole genome sequencing and array genotyping, and is offered at various costs based on the specific sample tested. In most cases, bioinformatics is required to convert the genotype file into a file suitable for upload to the genetic genealogy databases used in IGG. Sometimes the bioinformatics step also helps to repair the DNA sequence if there are any holes in the DNA from degradation or low coverage of an area of the genome. After the laboratory process is complete, the work of the IGG practitioner begins. So what happens next? The IGG practitioner works with a number of different outputs generated from the genotype profile and the databases used in IGG. Outputs from the databases used in IGG include a bioancestry report, also called the ethnicity estimate, and a genetic match list. Depending on the type of genotyping performed during the laboratory process, the IGG practitioner may also receive an estimate of the subject's mitochondrial and Y-DNA haplogroups. Each of these outputs is useful to the IGG researcher or team. The bioancestry report provides the IGG practitioner with a rough estimate of the subject's heritage. An example will be provided in the following slides. The genetic match list is the primary focus of the IGG practitioner's work. The genetic relatives of the subject will be analyzed by the IGG practitioner to form a hypothesis of how the subject is related to them. This allows the IGG practitioner to reverse engineer the family tree of the subject. The mitochondrial and Y-DNA haplogroup estimates for the subject allow the IGG practitioner to make an educated guess about the ancestry of the subject's direct paternal ancestors and direct maternal ancestors. This is an example of a bioancestry report from the same person from two different databases. This individual is believed to be half Irish and half American. Her ethnicity estimate shows that she is fully European. Keep in mind that these estimates are just that, estimates. They provide helpful context but are not the primary focus for the IGG practitioner. These are two examples of genetic match lists for two subjects. The match list shows a list of genetic relatives from the most closely related to the most distantly related. Relatedness is expressed in centimorgans. The higher number of centimorgans shared between the two individuals, the closer their relationship. On the left, the match list demonstrates the closest match of 70 centimorgans. This would be in the range of a third to fifth cousin match to the subject. If they are a third cousin, they would share second great-grandparents with the subject. On the right, we see that the top match shares 738 centimorgans with the subject. This is in the range of a first cousin. Perhaps this person would share grandparents with the subject. Which case do you think would be easier to resolve? In both cases, the IGG research team would build family trees for the genetic relatives to determine how they may be related to the subject. This allows the IGG practitioner to develop a hypothesis about who the subject is based on their genetic relatives. The hypothesis is the investigative lead produced in IGG. After the genealogy work produces an investigative lead, a potential candidate to be the DNA contributor of crime scene DNA or the unidentified decedent in an unidentified remains case, the IGG practitioner will share the candidate's name and key important information with the investigating agency. From here, the agency takes over and must further investigate the lead. In an unidentified human remains case, that may involve a proof of life search or contact with the person's family. In a violent crime case, this may involve looking into the criminal background of the potential perpetrator. Next, the lead must be confirmed with a means of confirmatory testing. In unidentified human remains cases, this can be an STR test from a family member reference sample, a fingerprint comparison, dental records comparison, or other means of confirmation. In a criminal case, confirmation usually involves DNA testing from a specimen obtained through a warrant or through surreptitious collection. It is only after the investigative lead is confirmed that a death certificate is issued for the formerly unidentified decedent, or that an arrest could be made in a violent crime case. This piece is important. IGG is a groundbreaking tool that can assist in cases of violent crime and unidentified remains cases, but it produces only an investigative lead, which must be further investigated and confirmed with secondary means. While IGG is a powerful tool, it is not a slam dunk that can solve every case. There are some barriers to resolving cases, and it's important for investigators to recognize that not every case can be solved with IGG. One potential barrier is ethnicity or heritage of the subject. The databases utilized for IGG contain profiles primarily from a Western European population in the United States and Canada. While there is some representation of other populations, cases involving subjects from minority populations may be more difficult to solve using IGG. Additionally, those cases might be impacted by scarcity of genealogical records. One example would be if the subject is from a rural region of Latin America. There may be genealogical records available in churches or municipal resources close to where the individual is from, but they might not be accessible online by IGG practitioners in other countries. An additional barrier is misattributed parentage. Families are complicated, and many families are impacted by adoptions or misattributed parentage events, cases in which the biological parents of an individual are not the documented parents. These can complicate the IGG search process, which relies on both the biological and documented connections between individuals. Thanks for taking the time today to learn more about investigative genetic genealogy. If you have any questions, please visit our website at ramapo.edu/IGG. You can also email us at igg@ramapo.edu.